Yeah, we hear the classic ding. We're going. We get back to where we were left off.
I'm trying to figure out. Yeah, I'm trying to figure out which one's the real.
Oh yeah, you're
Alright.
that makes
Oh, what do you mean? I'm gonna do it. Nothing's gonna do me.
So you we're going to begin. You guys are moving farther
I'm
back.
guessing this.
I've heard that before.
But I did that one.
Yeah. Come on in, join the party. Just getting ready to start.
That's actually what the FBI is going to start raiding.
They probably will. It could be trouble. All right.
I'm not trouble.
So this was, you might remember we were kind of around here, which these are the peripheral nerves, and it's an important point vis-a-vis understanding the individual, the distinction between the oligodendrocyte and the Schwann cell, but the consistent pattern of seeing these sheath gaps, which are we call nodes of Ranvier. So those are the six, and you need to know them where and kind of what they do. The six sort of varieties or cellular forms of the neuroglial cells, which leads us to the nerves themselves. And most of the nerves are of one type, particularly in the central nervous system. It's only a
Is that the statistics class?
no, not here. No, I don't know where it is. Maybe downstairs. I don't know where. Now, so and I've lost my train. So okay, so what you're going to see, and at least it's sort of as the rest of this goes on in this particular unit, you'll see that there are two forms that are pretty much exclusive to the sensory nervous system, and we really will explore them a little bit more when we look at sort of sensory nerve physiology. And those are unipolar, which you were just looking at an illustration of. That's a unipolar nerve. And the bipolar, which is very distinctive and usually found only in special sense organs. But the generic nerve is called multipolar, which means it's got multiple inputs and multiple outputs. So it can associate synapse with an immense thousands of inputs and thousands of outputs. That's the complexity of the system. You have so much going on for you to understand, to answer a question, to ask a question. All the you're being bombarded now with my voice, with visual information. You're trying to assimilate that, and whatever else you're doing at the same time, since we all multitask. So the nerves are the structural unit, very specialized. Obviously, they conduct impulses. They last your lifetime. I think they're almost exclusively amitotic. I think the exceptions are so rare, it doesn't pay to comment on them. And yes, it's all about oxygen and glucose. Your brain functions on glucose. That's why when we get to the endocrine unit, biggest problem with glucose is not too much of it immediately. They're both bad. Excessively high glucose will kill you, but it will kill you slowly. Excessively low glucose will kill you right away. That's a much more damaging potential. That's scary. And we'll get into all of that as well. All have a cell body, meaning, and again, that's effectively where the nucleus, but we don't use that term because it's used more than once, as you recall from anatomy. They have a cell body and they have one or more processes, which are either the transmitting or receiving arms of it. So the cell body, by convention, we refer to it as the soma, or the perikaryon cell body does just fine. That's effectively the nucleus of the nerve. Everything is made there. It has an abundance because it's generating a lot of materials, particularly to be excreted or to function in that process of exocytosing the neurotransmitter. So you see an abundance of rough endoplasmic reticulum, more so than smooth. And a lot of these substances, particularly certain neurotransmitters, are pigmented. So they are chromatophilic. And one of the names we associate them with is pronounced nacelle bodies. So they have a nucleus, nucleolus, pigments, etc. And obviously the very receptive area associated with the plasma membrane. Okay. So in most of the cell bodies for all nerve cells are located in the central nervous system. So there is a limited amount that you see will be associated with the autonomic nervous system and a limited amount with sensory nerves. And that, what you saw was an example in that unipolar nerve, which was looking at the Schwann cells, that those cell bodies cluster in ganglia just outside the, those are the dorsal root ganglia. That's, those are where you will see those sensory roots that you see both in the central and peripheral nervous system. They're associated. So we call them nuclei when they cluster in the central nervous system, but ganglia when they're outside of it. And again, sensory nerve ganglia are what we see them before they enter either the brain or the spinal cord. So the processes are very straightforward in that regard. And you've heard the terminology, I'm sure, before. Cell bodies and their process, PNS, neuron processes. The term we give to those processes, you recall from the anatomy, within the brain or the spinal cord are tracks. So remember, it's very standard. You remember commissural tracks go across, association tracks on the same side, projection up and down. Nerves are what we give to names of these accumulation or bundles that exit by way of the peripheral nervous system. And the processes are either receptors bringing information, afferent or incoming limbs, called dendrites, or transmitters called axons. Classic structure of a typical motor nerve that's there. This is the classic multipolar nerve. It has the cell body or soma that's there, the endoplasmic reticulum. The usual culprits are there, the nucleolus, chromatophilic substances. They have multiple projections that are dendrites, and each of the dendrites have multiple, and you'll see actual micrographs of them. So there's a tremendous amount of availability of information input. And by the same token, at the end of the axon, the terminals branch as well. So you can send a message, you can receive messages simultaneously from thousands and send messages to thousands. You can see roughly this area is where the axon starts. It's called the axonal hillock. It's a little mound. When you look at it micrographically, you can detect it. And that's where all of those impulses, if there's going to be an action potential, it begins there. And once it evolves, or once it is evoked, it makes its way down there very rapidly, particularly in a nerve like this. And the impulse is unidirectional. The flow of materials is in both directions. You have materials that are being in, but it's absorbing, because most of the neurotransmitters are reabsorbed either partially or totally. Materials are transported, and the bad news is viruses and bacteria, toxins can be transported. So it's a very interesting setup. So hundreds of these short tapering branches that branch again, same organelles as in the cell body. Again, when you're having the transmission abundances of mitochondria, this is a very energy dependent, of course. And remember, we use the term end plate potentials. Now we call these graded potentials. That's the distinction anatomically or terminology wise. So end plate potentials are only associated with a neuromuscular junction. Graded potentials are associated with strictly nervous system activity. And just like the end plate potentials, if they're insufficient to reach threshold, they only travel a short distance and they deteriorate. Okay. So again, what you'll see in one of these micrographs are all these little dendritic spines, these little ends that are accumulating inputs and information. So you can see it here. Here's a cell body. This is probably the axonal hillock. You can see it's thicker, more apparent with the staining that they're using. And then this is likely the axon that's projecting out. And these are, and you can see all these little spiky things on the dendrites that are there. The axon, conical area of the hillock, they don't all have to have them. We're going to focus on basically nerve to nerve communication, with basically chemical synapses at nerves. That's what our focus. There's a lot of variation in the nervous system. particularly in the brain, where there's inputs and modifications being made, where it's almost, it would be, like you hear something, the question is, I have an answer to that. Do I want to make that answer? Do I not? So it's almost as though there's subtle messaging that goes on, how I'm going to respond, to what degree I'm going to respond, with what intensity, with what rapidity, all those types of things. It's just, the brain's functioning like you and I are functioning. So some axons can be a meter long. That's the classic example of brain, let's say, going to the lower extremity to your foot, brain to the lumbar or sacral area of your spinal cord, and from that somewhere it's sacral area of the spinal cord out to your big toe. So the collateral channels are there, profuse branching, axon, we call them terminals, terminal. Some people say buttons, it's pronounced bouton. The same. It's French. My favorite saying in French, by the late great Julia Child. Bon appetit. That's another HBO show you gotta watch. The lady who does that is a wonderful actress. She does Julia Child better than anybody I've ever seen. Just great. That was like an 8-show arc. It was just fabulous. The axon conducts the impulse. We know that. It generates them along the axon's membrane. They call it fancy the axolemma to the terminal. The terminal is where everything is just like the terminal going to a muscle. Okay, they're released the neurotransmitters, in this case, into the extracellular space. It happens to be at a synaptic junction with another nerve, and it's going to send one of two messages. It's very straightforward. It's kind of like a computer in the sense, yes or no. Okay, the early computers were basically as a binary function. So these are either going to send, and most of these nerves secrete more than one neurotransmitter depending on the accumulation of the signals. It'll either tell the next nerve be more likely to fire or less likely. So more likely they will excite, less likely they will inhibit. And again, the fact that you're inhibiting a nerve doesn't mean you may be making it less likely to fire. The end result may not be inhibition. What you're going to be indulging in on the weekend, you're going to be you'll be ingesting a disinhibiting agent.
All right.
Alcohol is a disinhibitor. So it will take your normal sedate behavior and potentially that will inhibit that, and you will be dancing on a table.
I'm built differently. I don't know, Dr. Stein.
Hey, there's nothing wrong with dancing on a table.
Heck, I'll dance on the ceiling if you want me to.
Thankfully, my daughter is in Cancun this weekend because I'm afraid you would run into her.
You're saying I got that W Riz.
What?
No, then you carry on. It's good. Is this recording?
Yes.
Can we clear that off?
In the words of Sergeant Show, I know nothing. I see nothing. I know nothing. I didn't. We will only distribute it to you people.
You set me up, Dr. Stein.
I did. I didn't, but I did, I guess. Okay, so all of that tough conversations with you. How are you doing today? Well, I'm having a good day. Nobody's excited me all day. Okay, and renewing proteins, quickly decay. We're going to get into that nugget as well. The axons, internal. This is the important part. It can be anterograde, away from the cell body, or retrograde. So move along the axon. So there's a whole series of internal tubules we call neurofibrils. What's interesting in Alzheimer's, they get tangled. That's why we think it's a protein malfunction that a lot of individuals think may be related to prions, which you might remember from micro, and for those guys in MCAT, we talked about them a little bit, because we do see the same kind of damage in Alzheimer's as we do with prion-based diseases. And we know there's about 5% of senile, what's called SDAT, senile dementia of the Alzheimer's type, that's related to prion, directly related to particular types of prions that they can identify. So mitochondria, cytoscalar, all the construction materials, membrane, etc., as well as passageways for the neurotransmitters. Retrograde organelles that are going to be recycled, some signaling molecules that are going to be reabsorbed and repackaged and recycled. Bad news, viruses and bacterial toxins. So it's a passageway. And indeed, that's the distinction. When we start looking at certain types of toxins that work very rapidly, neurotoxins is the subcategory. I do not know whether you covered that in micro or not, but that is one of the subcategories. So certain kinds of snake venom are neurotoxins. Cobra venom is a neurotoxin. In our area, the 4 types of venomous snakes. I don't want to forget the Gila monster. Why would I? Okay, are copperheads and coral snakes, and the other water moccasins and rattlesnakes are hemotoxins. Just a little toxicology wouldn't hurt you. So that's part of it as well. So polio, rabies, herpetics, okay, tetanus toxin, things that you've touched before. And again, this is the idea of basically implanting viruses using retroviruses, basically taking the HIV out of HIV and using the reverse transcriptase enzyme and basically that's what gene therapy is about. Okay, microRNA, etc., those types of things. This is the myelin sheath. Myelin is very important. Myelin is a protein and fat material. So it's very unique material that's a wonderful insulator. So it's protective and insulatory, and it increases the speed. And you'll see it in the animations probably better than I can explain. And again, I touched on it before. We used to think the signal would like jump on the outside of the nerve. It doesn't do that. But we still use the same name as saltatory conduction or jumping conduction. And we know now that where you're going to see those voltage gated channels, similar to the ones we talked about in muscle, those paired sodium and potassium channels, are really abundant in the areas in the nodes and fairly absent where it's covered by myelin. So the signal is being transmitted within the nerve very, very quickly between those 2 areas. And really the jump is from node to node, but inside rather than outside. If you want to think of it that way. So the more myelin, the more nodes, the faster the signal. So you're going to see increased amounts of myelination, increased thickness of nerve is directly proportional to the speed of transmission. So myelinated, and we even characterize them as different degrees of myelination. So myelinated it depends on where they are. The myelination has nodes. It's a little bit different, as you saw before, in the central nervous system with clusters of white matter forming myelinated tracts, even though they have, it's architecturally a little bit different versus non-myelinated that are there. So in Schwann cells, and this is where the distinction is, as it wraps around the individual cell, it has like a little bump because the nucleus is inside of it. So it's what they describe it as jelly roll fashion. I personally think of it's like making cinnamon rolls and flipping them on their sides. They're no fun. So one cell is one sagment. They have perinuclear cytoplasm, and et cetera, et cetera. You'll see the illustration better than I can explain it. The plasma membranes, it's mostly fatty with a little bit less protein, so you don't have the channels or carriers, electrical conductors. They interlock very, very well. That's what I mean. That's sort of the bump. And you can see it just rolls and rolls and rolls around. I like that. It's very, very rolling it. Sicilian cooking, they roll everything. Look. Roulade of beef and nobody cares. So you can see how proportionally thick the sheath is compared to the axon itself. When you look at this 24,000 times, so you're looking at an individual area within an individual axon and look at the girth of the sheath. And they even will show you some of the perinuclear cytoplasm of the Schwann cells that's there. So it's a good illustration. So again, I always refer to them because this was learned nodes of Ranvier versus sheath gaps. It doesn't matter. There's also collaterals can emerge. So you can have contact in those gaps going in, input and output. The non-myelinated fibers are considerably thinner. Okay. And as you can see, maybe you have one cell surrounding it. It doesn't do that jelly roll coil up. The same illustration you saw before. So in the central nervous system, it's about the oligodendrocytes, as you recall, not an individual cell per se. It's about the process emanating from the cell. And you can see it can wrap multiple axons, space limitation, if you want to think of it that way. Sheath gaps are present. They don't have that outer collar. Thin fibers are unmyelinated. But again, adjacent neuroglia do play a role with regard to that. So in the central nervous system, you already had this in anatomy, white matter versus gray matter. White matter, myelinated fibers, the tracks. Gray matter, pretty much everything else. It's more or less synapses, cell bodies, and certainly non-myelinated fibers. But so when we see the cortex area in the cerebral area or the cerebellar area, okay, that's the gray matter that you can see there, or certainly those deep islands of gray matter. And you saw that illustration as well. These are the different structural classifications. There isn't a whole lot left in this particular segment, PowerPoint segment. The multipolar, where it has multiple processes, one axon and a bunch of dendrites, just the big player. Bipolar, comparatively rare. It allows, and it's something we're going to explore, particularly when we do the special senses, when we look at the physiology of hearing and the physiology of sight, that you're going to see this is the transition of the signal from taking an optic in the eye, from taking a visual signal, basically a reaction, a photoreceptor, and being able to transition that into chemical changes and then transitioning those chemical changes into electrical changes. So it's like in the old days, we used to say it was like we have two sensory nerves in sequence. Because originally everybody thought, well, it's sensory and motor. And now the difference is that you have a sensory nerve that's processing a sensory signal into a different kind of sensory signal, effectively creating signal transduction, changing the nature of that signal. So they're relatively rare. You'll see them again, mostly abundantly in these areas. And the unipolar is the classic sensory nerve, light touch, hot, cold, sharp, dull, pain. The peripheral process is associated with the receptor. It makes its way dorsally back to the brain, or the dorsal portion works its way back to the brain or spinal cord, but it forms a ganglia, a cluster of those cell bodies. So here there's a distinction. Typically the cell body, the dendrites lead into it, and a long axon and a multipolar. Here, and they're very, very tiny. The length is not nearly the same. Is the cell bodies within the middle, and then you have a dendrite on one side and an axon on the other, depending whether it's a sensory or motor-based signal. And here, for instance, the peripheral process, this is a receptor. Yeah, I touched something, I felt it. Cell body is outside before it enters the brain and the spinal cord. And this is maybe a little bit better view that we see in retina and olfactory. For instance, these have an interesting name because the cerebellum has a tremendous amount of synapses. Everything we do is constantly being monitored cerebellarly, to make a word, and recording that information. Any motor, I remember, if you remember what the cerebellum does, everybody, I really focus on that more in the anatomy portion. It's ipsilateral motor coordination, moving on the same side. So every time I do something, it's like a little pattern that stores in the hard drive. And the hard drive is those kinds of cells. Purkinje is the name of those cells. Purkinje was a big name. We'll talk about Purkinje fibers in the heart. Maybe it's the same Purkinje. Who knows? And the pyramidal cells, again, these are again part of that pyramidal area are cells that are being associated with the main motor tracts directing us to do motion. So how we classify them: sensory impulses from sensors, pretty obvious, nearly unipolar, ganglia outside, cell bodies within, and cell bodies and are located within ganglia within peripheral nervous system. Motor impulses to whatever the effector is. Nearly all, not all of the cell bodies are located in the central, because when we do the autonomic nervous system, you'll see that. And then by far and away, 99%, it's not input and output, it's all about processing. 99% association or interneuron. So it's really how we interact is on a basis of that way. Even the simplest reflex has at least one interneuron. So they're always present. I guess that last little slide. So here you're seeing motor going, to a motor cell. Here you can see, and we'll explore this in the eye, and here you see, and that's the ganglia I was referring to. And I mean, the most interesting thing is that's where herpes viruses lay dormant, and why we follow herpetic problems along the lines of sensory nerves. So we all done with that, and we can start the next one. Part B. This is the electrical activity nugget that's there. So the part B talks mostly about, this is the electrical nugget that's similar to all the stuff we talked about at the neuromuscular junction and the cross-bridging site, all that stuff. The last section deals a lot with neurotransmitters. Those are really the ways this chapter is divided. And to me, I think the neurotransmitter one is the most interesting, because I think it really focuses on the synapse and the events that occur there. But this is important as well, because this is really the electrical activity. Arresting membrane potentials. Nerves more rapid in their change. They're very excitable. This is physics. And as you all recall, graphically, I don't do physics. Wherever he is, does physics. Nice guy, British accent, Liverpool fan, but can't have everything. We'll probably meet up for the game on a Saturday. The Saturday you guys start break. Our teams will interact with them. So opposites attract. Yeah, we know that. Energy makes membrane potential, and energy is liberated when the charges change. Got that? Yeah. And we measure it in volts. That's all I'm talking about. Okay, we're not talking about current. The interesting about the resistance and the insulation is where the myelin comes in. Okay. So demyelinating diseases, which I'm sure I commented on, the most famous of the demyelinating, there's two. One is presumed to be autoimmune disease, that's multiple sclerosis, which can occur centrally and peripherally, and the other one is a autosomal recessive disorder that you might recall occurs mostly in the Ashkenazi Jewish population, read that as me, Central European Jewish population, called Tay-Sachs disease. Those are the two biggies. Okay, and I'm not doing Ohm's law, I'm doing this. Same thing. Large proteins, just like they are in any other area of a membrane, we looked at cellular membranes, serve as selective membrane ion channels. Potassium, these are the leakage channels. Okay, potassium channels, sodium channels. So you have leakage channels, and of course for potassium, you're also going to see gated channels. So the leakage channels, which we know are always open, little pores, the gated channels change shape, open or close, either in responding to a ligand, a chemical gate, a voltage, an electrical or voltage gated channel, and mechanically, like in the ear, you see a mechanically gated channel. So these chemicals are very specific to the type of neurotransmitter. Voltage gated respond to changes in membrane potential. Mechanical physical deformation, pressure. That's that eardrum vibrating. And you're going to see some very familiar faces that are here. Okay, so we've seen most of this before, whether it's a chemically gated and the ligand, in this case a neurotransmitter, attaches. Again, many of these will be of the sodium and sodium out, potassium in kind that are there. Voltage gated, having to do, as you can see, the rapid flip flop in the membrane change, opening the channel. When gated channels are open, ions diffuse in quickly along their gradient, higher to lower, and electrical gradients go in the opposite, obviously toward their opposite, positive to negative, etc. Back to the resting membrane potential, not a whole lot different than any other cell. Those leakage channels, principally the potassium leakage channels that you remember from not too long ago, create that electrical. They really are the linchpin for resting membrane potential. And of course, that creates polarity. And we review the distinction in the ionic composition between the intra and extracellular fluid and permeability differences. So it's really a review of what went on before, and they show you the same diagrams that are there. The big players, the extracellular has a higher concentration of sodium than the intracellular. Okay, the intracellular is potassium. The balance that offsets that globally within the cytoplasm or outside of it, keeping it electrically neutral other than along the membrane, is chloride outside, the negatively charged proteins inside. Going to see chloride playing a bigger role. That's coming when we get to the neurotransmitters. Chloride channels are big players in the nervous system. I see it. Look. Oh, I know how loud it is because we were on a video link in.
The human brain alone contains about 100 billion nerve cells called neurons. A neuron, like all cells, has negatively and positively charged ions in the fluids located both inside and outside of the cell. However, the ionic composition of these two fluids is markedly different. The inner surface of the membrane is negatively charged, while the outer surface is positively charged. This unequal distribution of charge creates a voltage across the plasma membrane, known as the membrane potential. In a resting, unstimulated neuron, this membrane potential is known specifically as the resting membrane potential. The value of the resting membrane potential depends on two factors. The first is the presence of concentration gradients across the plasma membrane, especially those gradients for sodium and potassium ions. The second is the permeability of the plasma membrane to sodium and potassium. In the resting state, this permeability depends on the number of non-gated leak channels present in the membrane for these ions. The magnitude of the resting membrane potential, inside relative to outside, is measured in millivolts using a voltmeter and two electrodes. We'll now place both electrodes in the extracellular fluid. Note that the voltmeter records 0 voltage. This is because the unequal distribution of charge exists only across the plasma membrane. The intra and extracellular fluids are otherwise electrically neutral. However, when we insert one of the electrodes into the intracellular fluid, a voltage is recorded. The value of the resting membrane potential varies, but on average it is minus 70 millivolts.
Okay, so we're good, and we knew, and it's review anyway. So, and we see how it's generated, et cetera, et cetera. So, all of those things. The permeability, slight, 25 times more permeable with high white potassium is so important, and chloride can move. You'll see the role that it plays down the road. And so they're just, and again, we constantly, just to maintain that, even after subtle changes, is the continuing role of the sodium potassium pumps that are there, restoring the conditions. And now we'll begin to see a bit more of what's going on here with regard to the changes that are associated. As we really will start to see it. And it's too hard for me to go over those distinctly. And there's another animation that's pretty good that's coming up. So, I mean, I like the way these are done. So it changes with the concentration of ions or permeability. And again, graded potentials versus an action potential. Not end plate potential, but graded potential. Okay, and it's really the same, short distance versus long enough, a signal that's sufficient to go to the next level and trigger the voltage gated sodium and potassium channels. Okay. So the terminology we use with these graded potentials is called depolarization and hyperpolarization. It's a subtle difference. So when a neurotransmitter, a tiny amount is released, it may only create a limited potential, a graded potential that goes toward making it more likely to fire, moving the resting membrane level closer to zero, making it more positive or less negative. So you're more likely to fire. So we call those excitatory. And really, it's the summation of all of these. We didn't have that in the neuromuscular system. Everything was excitatory. It depended on how many. In this one, when the nerves are conversing with one another, effectively sending signals, it's basically a fire, don't fire. Excitatory means fire. Inhibitory means don't fire. And we call that hyperpolarization. Because what it's doing is it's allowing chemicals in or out that are making it less likely to fire. More negative or less positive, depending whether you're an optimist or a pessimist. Glass half full, half empty. I'm an awful optimist. If you're ever not in a good mood, I'm one of those. I'm a happy person. Other than when I have to come here. Oh, don't start. This is my happy place on earth. Being with you, because you're such a fun class. Well, most of you.
You're looking at me.
No, I'm not. But gotta pick on somebody. And it gets tough. I pick on Chelsea all the time. I have to give her a break.
That's true.
It's true. So more likely to fire, less likely to fire. Depolarization, hyperpolarization. So they're a very short lifespan. The stronger the stimulus, the more voltage changes and the farther the current flows. Okay. So we can get to that. Okay. So depending on the stimulus, we're going to open a certain amount of ion channels. Now, according to the location and function, we have receptor potentials, graded potentials and receptors of sensory neurons, postsynaptic potentials. So you have different types of terminology that are there, and you'll see them. So here's the depolarization, very limited. You can see from the and everything as you look here. This is the important distinction. With muscle, when it was stimulated, it went in all directions. This is only going downstream. It doesn't go backwards. It's the nature of this stimulus. When it occurs, it only goes downstream. So depolarization spreads from one area of the membrane to another. So even though you're seeing some localized changes that are here, it's somewhat limited and then it decays. Current flows but dissipates quickly. Graded potentials are only short distances. So it starts here and you see it declines over distance. Long distance, muscle cells, axons of neurons, brief reversal, action potentials, the name that we give them, and they don't decay. They're self-perpetuating. So another term you hear is a nerve impulse. That's there. Specific gated channels. And now, and it's all about these guys again. Then that's going to play a role vis-a-vis the refractory period. It's very limited, but there is one. So when that activation gate swings into it and deactivates it, it creates an inability and we know how rapidly you can fire your nerve. But there still is an element, a finite element, even if we're in fractions of a second, where it can't fire. So what do we see? All gated sodium potassium channels are closed in rest. Only the leakage channels are open, and they're being offset depending on the degree of leakage by those sodium potassium pumps, maintaining it. So each sodium channel has activation and inactivation. When we get to the voltage gated channels, activation gates, closed at rest, open with depolarization. Inactivation gates, block. Open at rest, blocks the channel to prevent more sodium. Potassium, open or closed. So voila. So we see what happens here. Normally this area, here we are at the resting state. At depolarization, the local currents open the voltage gated sodium channels. Sodium rushes in, activation, inactivation gates open, more depolarization, etc. At threshold, what will happen, it creates a positive feedback loop. Bye guys. Bye guys. Unlike you, they like me. But they're young. They're young. At threshold, usually around minus 55, just like it was in the muscle, large spike, big jump. So we're actually generating, a significant change voltage-wise. That's there. So you can see what happens. That inactivation gets out of the way. Repolarization, the sodium channels inactivate. The potassium channel, if they are technically closed, they're blocked. Potassium channels open, membrane permeability of sodium down, action potential stops rising, and now it immediately begins to decline. And you see in nerves, it's a very, very rapid, distinctive spike. Okay, so potassium exits, repolarization, membrane returns to its resting membrane potential. So you see here, it looks like it's open, but it's blocked. Here, everything's exiting out. What's interesting, as the sodium channels reset, there's a tiny amount of what's called hyperpolarization that occurs because the potassium channels remain opening and allow excessive potassium to go out. So it actually overshoots. And we see this a lot biologically. You see a lot of overshoot in a lot of systems. Even when hormones are released, you'll see a lot of activity, and there's mechanisms to prevent that. The one we'll talk about the most is the insulin glucagon relationship in endocrine. So it hyperpolarizes as the sodium channels reset. So now it's swinging away. The reset closes the gate, but we begin to see this finite area. And then they just go over those once again to show what goes on. So listen to this as we get ready to conclude.
Neurons communicate over long distances by generating and sending electrical signals called action potentials. Most action potentials originate in the axon's initial segment, a region also called the trigger zone. During an action potential, voltage-gated sodium and potassium channels open and close, thus altering the membrane's permeability to sodium and potassium ions. As these ions diffuse through the channels, the membrane potential changes. It depolarizes from the resting state of minus 70 millivolts to a peak value of plus 30 millivolts, and then quickly repolarizes back to minus 70 millivolts after a brief period of hyperpolarization. In this topic, we explore, step by step, the events that occur as an action potential is generated in the axon's initial segment.
But aren't you going to do it? Oh, it's terrible. Okay, so repolarization is the reset. And if you look back, just real quick, the distinction is here where it starts. I'm trying to find I saw an illustration that might, let's take a look. When they come up here, this is where the action potential is generated. Back here, there's no voltage-gated sodium or potassium channel. It's anatomical. When the membrane changes, it contacts them here, starting the process. That's why it's unidirectional. When you muscle, that neuromuscular junction is in the middle of the muscle. It goes in all directions. That's it. So it's an anatomical distinction. And here's the repolarization. The sodium potassium pumps go crazy. And not all depolarizations result in action potential. We know that. It must reach the threshold. It has to be 15 to 20 millivolts typically for all the permeabilities and to generate. So it's very much of an all or none phenomenon. Okay? It's either going to fire or it's not. There's no partial degree of it. It's not like the analogous to the twitch and all that. It's not quite like that at all. And it propagates, transmitted from the origin down the length of the entire axon. Sodium influx through voltage gates in one area, open, and it leads to the next area and the next area until finally, just like we saw at the neuromuscular junction, the impulse gets there, hits the voltage gated calcium channels, get the synaptic vesicles fused, exocytosis, etc. So non-myelinated axons, each successive segment, in myelinated, it differs. Since sodium channels are closer to the AP origin, are still inactivated, no new action potential is generated there. So the action potential only in a forward direction. It's only going that way. And they're just showing you what's happening with all these illustrations. And I'm not going to do this one. We'll start with this and come back on Monday. Okay? Thank you. That one's the most interesting one.